Thank you, everybody, for sticking through 2 long weeks of CompuCell workshop and then 2 long days of hackathon. Like I said, we've been very impressed with everybody's fortitude and with all the time and energy that you put into everything. So we will have our brief final report presentations here, just so that everybody has a chance to kind of show us where they ended off. Just as a reminder, this session is being recorded and live streamed, like other sessions, and will be distributed on YouTube after the fact. Sorry. And again, the materials for everything that we did today will be found in the hackathon folder moving forward. And as well, if you would like to use the Slack as a way to continue to communicate with your team after the hackathon, that will be left up for at least the next year. Typically, after a year of the Slack being open, we start to lose some privileges and features, but the room itself will still be open for chat. But I think that after a certain time, joining becomes more difficult. So I would definitely recommend joining now if you're planning on using that as a way to stay in touch with your group. So like we said, for final presentations, and again, these can be very informal. There's kind of 4 things that we want to hear a little bit about from each group. We'll say a maximum of, well, we'll say between 10 and 15 minutes, shoot for about 10. I think people are tired at this point. But just give us a very quick overview of the biological background of your problem. We've been talking about our simulations mostly in terms of the output you've gotten so far, but I think it would actually be useful if everybody just spends a minute to remind us of what it is they were trying to model in the first place. And then you can show us your model, or at least the condition that it's currently in, show us the simulation running. Take a brief minute to discuss a couple challenges you faced along the way this weekend, and maybe a little bit about how you overcame them. These kinds of things are very useful for everybody to know. So in other words, what plans did you have to change in order to get your model to where it is now? And then the last thing is just summarize your plans for future work on the model if you are planning on continuing to work in the future on the model that you started building this weekend. And that includes what else still needs to be done or added to the model in order to answer the biological question you were interested in, and what kind of help might you like from us moving forward. So I think with that, and I have our people slide here again. I can display this at the end if anybody needs email. Yes. All right, we'll do reverse order. We'll do Marcella and then Cesar, and then we'll work backwards from there.
I'll be really quick since I have my 2-year-old with me. He's not going to allow me to say much, but I can quickly share and just run and say, for me, I wanted to just start building really simple submodels that I could put together to build a general, like an inflammation model, and focusing right now just on macrophage differentiation from pro to anti-inflammatory phenotype and back again in response to some kind of stimulus. And what I have here is just some M1 and M2 pro and anti-inflammatory macrophages that secrete cytokines that transition to different phenotype based on their exposure to the cytokines. And so I have some plans at this point to, like right now, there isn't any inflammatory stimulus, but that should be what's driving the secretion of the cytokines. So I have plans to add that, and some plans to maybe make a teaching demo out of this. I think would be a really nice thing to do. I'd like my students to work with this, and then just keep adding other components.
That sounds pretty good. If you would actually like to talk in the future, I'd be interested in talking more about developing the teaching demo.
Great.
And we have Giuliano wasn't able to give his talk this year, but he gave a talk last year on publishing these things to NanoHub. So if you did want to develop something that could be launched with basically one click that your students could investigate, we can get you the resources on how to do that.
Oh, great. I will be following up about that then. Thank you.
Yep. Julio, that makes you next.
It's me. Okay. Hello, everyone. So I wanted to share my simulation I tried to do. It's called Semiconductor Forms, and actually, this is the image. This is the phenomena, the image that I'm trying to reproduce. It's certain materials, when irradiated with heavy ions, they start to grow like foams, and there is no theoretical model that explains everything that happens when the process is being executed. So simulation with CompuCell would be amazing to start thinking on what's happening inside. So the event description, what is known already is that like fast ions, they travel through the material and they increase the temperature locally, and has a chance of a bubble starts growing, and then it rapidly solidifies again with a different structure. So, okay, for the simulation, I had some assumptions. I had just one big cell of solid, I called it solid, and it has a constant target volume because mass conservation is a true thing in this process. And this is a square, not a circle. And the next thing, every couple of MCS, a random pixel inside the solid gets chosen to grow a bubble, and it starts with a pixel and has a really high lambda volume, so it grows really fast and keeps there. Before getting another pixel chosen, and I turned the old gas material to a pore material. So why I decided to do this, it's because when I have different cell types for these two parts of the process, I can change its lambda volume or its target volume as I want without setting it to medium like I was trying to do before. So another important thing is that when there is no irradiation, the solid must keeps its form. It was very interesting. I'm going to show first at least the beginning of the simulation. I did it with a kind of big lattice, so it's very slow to keep to 50,000 MCS. So what you can see here is that bubbles are getting created with green, and then they almost instantly in the simulation turns to purple, and then they like pores. And if you watch carefully, in the final I adjusted the lambda volumes, so they almost look like circles. So it's a great thing indeed. And okay, it keeps growing, and the results for many MCS that I had were like for 15,000 MCS. In the calculations I did, it was about 1,000 ions passing. You can see many bubbles, and without the cell borders, it looks more naturally. Like you start to see pores forming, And when you go up for 30,000, you start to see like something getting shape. And it's good to know that it's very different when you do for big bubbles to small bubbles. When you grow your lattice and keep bubbles small, the shapes do not go so wrong. By wrong, I mean like a big bubble inside material occupying all the space. And okay, when you grow to a lot of time, like 3,000 ions, you see something like this. In the last picture, I just made the pores black to look like something you could compare with reality. And you can see it's not perfect, but you can, like, closing your eyes and punching your head a little, you can actually see something similar here. There are different types of porous structures or foam structures in experimental in experiments, and you can see from rounded bubbles to more advanced structures like this one or even this one. And this is like a mix of all of them. But I think it was a good result for a first approximation. I did very, very, like, many simplifications to do this. And the problems or the possibilities of getting things better from this model would be implement something that I could distinguish a material from another, because this event does not happen in every material, so it's just specific materials. There's something that would be very much better if I could have local volume interaction, because when we see like this, I have an ion track and another bubble that has been formed many MCS ago. They still get, reshaping, and that's not the point, because I have, like I said in the beginning, I have local temperature change, and so it does not make sense to change in the other side of the structure. There's something with the typical ion penetration length. I used like uniform random Y to have a bubble, and it's proven to be not very good, because when you do this, since the material grows with time, you have much more bubbles concentrated in the bottom, and sure, the simplification is shown right here. Otherwise, you can see in the experiments that bubbles grow like uniform from the top to the bottom, and it's not what happened here. And another thing that I have seen is that it must have something like a law behind everything or something like that, that can be done in CompuCell, but I think it comes better when a theoretical model exists behind that, reproduce the shape of the structure much better. As you can see, I have random shapes of bubbles getting together, and it's not what really happens in reality. You can see that it has like a real structure or something. Jim told me about like trabecular bonds or something like that. They look really similar to the reality, more than the simulation I did. And it appears to have something that applying the simulation would have a really better final shape to the structure. And the final thing that could be done right now, but I just noticed in the end, is that bubbles that have contact with the borders should not have the pressure like the ones that are confined. I mean, they should be like medium when they enter in contact with the borders or the top of the simulation. And that's all I had to say. I would like to thank you all for the workshop. It was like amazing 2 weeks, and these are my 2 cats. And okay, that's it. Thank you all.
Okay, so for those who listened to my original project presentation, I was going to make a model that I still have to do, and that's the whole reason for me being here, about the biological phenomenon I'm working with, I'm studying. The model I wanted to do is one that replicates this particular setup. These are bacteria, the bacteria I work with, called Myxococcus xanthus. They move along, they are here tile colored spaghettis. And they move along these yellow trails. And as they move, the trails change their behavior. They move faster, and they prefer to move along those. And as they move, they create more and more, and they create these huge networks of trails. However, at the very beginning, I understood that it will be, for my skill set, too complicated to attempt this in 2 days. So I'm just revisiting this original idea in case further on I do need help, so that the instructors sort of remember what I was originally going for. But since I decided that it was going to be a bit too difficult for me to implement now, I decided for another project that I had in mind for a while, that essentially to replicate the architecture of biofilms. Biofilms are these clusters of cells that attach to a surface, and they, depending on the interactions with the medium, they become very dense, or they create cavities that allow the flow of medium, or prevent different phenotypes of bacteria to be exposed to, say, a low oxygen medium, for example. So phenotypes, depending on the phenotype, it's their location within the overall biofilm. So that simple idea is what I wanted to implement, and essentially this is it so far. The idea was to have a bunch of cells, scattered bacteria, that were essentially represented 2 phenotypes. The green ones were susceptible phenotypes, and the blue ones were resistant phenotype that was able to tolerate more on the external medium. So what I was going for is to see exactly this architecture, where this layered architecture, where the green phenotype was sort of protected from the medium, from by the blue ones, the resistant phenotype. And for this specific project, well, I need a couple of things. first I need to solve the architecture, the shape of the cells. That specifically is something that I really need for the original project to be right. So it proved to be a bit too difficult because, as Pedro told me, if you have a ratio, a very thin cell, it ends up breaking, and that's something that I continuously was experiencing. So I need to get that right for the model to be realistic further on. And for this particular setup, I would like to implement more phenotypes and play both with, say, bad mediums, with mediums that are bad for the cells and mediums that are desirable for the cells, and see how they aggregate and create different architectures and stabilize around that. And yeah, so it's a very, very simple model. My skill set is so far not very good, but this workshop has been very, very helpful for me, especially because it helped me, overcome the fear I had to CompuCell and programming, etc. So I feel way more confident now to keep building my models from now on.
Okay. Hi everyone, I'm Shilai. I have been working with Fatemeh working on lever model in response to acetaminophen. So she's been working on Morpheus, and while I was working on CompuCell 3D to reproduce the results. Let me share the screen. So this is the expected behavior of cells inside a lever lobule with the hexagonal shape. On the vertices, there are portal veins there. It's the place where acetaminophen come inside and diffuse inside the lever lobule. Well, there is a complex metabolism that triggers some chemical inside, like at the central area, which will result in necrotic state of hepatocytes cells. And this is what I did so far with helps from James and Jim. So I spent a lot of time, honestly, just working on the initializing the cell fields in CompuCell 3D. I manually draw a hexagon inside Python and use Latin hypercube sampling to randomly assign cells inside this region to make sure that every single grid will has at least one cell, and then let it relax a little bit to fill in the gaps. And then we got this hexagon shape. Also, I did some parallelization inside the initialization to treat the coordinates as a tensor and use a rotation matrix to duplicate some simple geometric shape to produce this hexagon shape. And the acetaminophen, the diffusion of acetaminophen inside the liver lobule will trigger some metabolism of hepatocyte cells to produce some toxicity and also some metabolism of detoxification, I guess. And this is how the acetaminophen diffuse from the source of portal veins on the outside. And here there's a toxin level, I guess, inside the cell that can result in the necrotic states of hepatocytes. And if we run this model long enough, we'll see some cells dying from inner circles. Yeah, that's pretty much it for our project.
Hello. Okay, I wish that I could give a nice polished talk like the rest of you, but I was working until the very last second. We were working because Maria came back. She had an internet problem and Pedro helped and Joel helped on and off. So remind me if there's anything I'm supposed to say, but you know, I'm working on trying to get an evolving system. Did not get that far, but I have been working on getting something that should be evolvable once I get there. Much of the weekend was spent, as you all know, struggling with adhesion flex, but I think I'm getting on top of it. And this last hour was a lot of fun, finally getting things that are wrong in interesting ways. And I'm going to show you a couple of images. I guess I need to share my screen. I don't have a PowerPoint for this. Now, one of these says image 1. Okay, you should see this. I have predator and prey cells, and they're using adhesion to the predators are increasing their adhesion molecule when they encounter prey and trying to engulf it. And I also added a one of these tracking fields to show the level of adhesion molecule in the cell. So you can see these 3 that are grappling with prey cells have high adhesion molecule. But there was a problem with this if I let it run longer, which is image number 2 here. Image number 2. So after a while, if two predators touched each other while both of them are touching prey, they would both have high adhesion molecules, and their surfaces would blow up and they would freeze in place. And actually, this has a lot of really interesting, there's a lot of really interesting reasons why this won't be a problem once I have real evolution going. This won't happen under the circumstances where you have energy constraints. So even increasing your adhesion molecule costs energy. Fighting with another cell over prey will cost energy. There are all kinds of reasons why this won't happen. But for a short-term fix to get this improved, I realized that, so we had the incomplete engulfment that you see down here, which is this one. And if I fix the energies so that I could completely engulf the cell and eat it and destroy it, then we would never get to this situation. And so I was able to do that. And so here you have cells that have actually engulfed their prey. And then the last step was I wanted to kill the prey once it's engulfed. And my code is to detect the engulfment and then knock off the prey. And I for some reason, I don't have it quite working so that they get inside the predator and then disappear. But what I do have I have them sort of instantaneously disappearing. You no longer see them. I can actually run this for you from scratch and place them next to each other so we don't have to wait as long for them to start interacting. So what you'll notice is the prey disappearing as the predator picks them off. So they never get into that trouble, but I wasn't able to get it right so that it has to, there's some anomaly with neighbor counting, which is what I was using to detect engulfment. So I'll get it looking better later, but that's what's happening. So we have some behavior here that has really a lot of potential for being evolvable and causing a very complex, rich set of interactions. So it's not evolution yet, but I'm happy with it for 2 days. And I definitely do plan to continue this project. It's going to be a weekend project because this is not what I get paid for. So, but I will definitely take this forward. And I think Maria is interested in continuing with me, and I suspect Pedro and Joel might have some interests as well as I go forward. And I'm in their labs with them, so. Something else I should have said, Hayden? In terms of
I think that about covers it.
Okay.
I mean, the other question is what help you need, but we'll be in touch, I imagine.
Yeah.
All right, thanks, Sharon. Next, we'll have Pete go.
Oh. So I hope it's all right if I take quite a while because it's been a while since my last update, and I'm going to start off by reviewing some of the biological background. So my project is about B cells migrating between the dark and light zones of a germinal center. And you have a few cell types that are involved in that, some stromal cells that are in the background, the B cells themselves, of course, which have two subtypes, central blasts, which are the proliferating ones, and central sites, which are the ones that migrate towards the right. You also have the T helper cells and the Fyffe dendritic cells that have the long arms. So one of the things I'm going to cover in this presentation is how I adapted what I saw in the literature to my own model. So I ended up discarding the idea of stromal cells, partly because they have no function, but mostly to improve the performance of my model. Besides that, I've also scaled things around a little bit, so these were not quite proportional to another model I saw, which you can find up here. So the hypothesis I wanted to test was, how does the level of antigen that a B cell picks up every time it visits a Fyffe dendritic cell get replenished? And how does that amount of replenishment affect its performance in the selection algorithm? So the way I implemented this, which I'll get into more detail later, is if a B cell has a very high level of affinity, then it'll gather more antigen during its visit to the light zone. And thus, because it has more antigen in the dark zone by the time it returns, then it will have greater amount of proliferation. So here I've discussed a little bit of the algorithm in more detail. So a B cell has two receptors, an MHC class 1 and a class 2. So let me get a wizard pointer. So what should really happen in the model is the dendritic cells, as you saw earlier, have these really long arms that can reach all the way up to the B cells in order to distribute the antigen. For my model, I've simplified this a little bit and just had it on contact and made the cells kind of large, which you'll see very soon. But if I wanted to extend this model further, I could do that and just measure the distance using a fast calculation algorithm. So every time a B cell touches an FDC, it should have some opportunity to gain a little bit of antigen every Monte Carlo step until it reaches a certain threshold. So this is something that I think I would like to research more into, because other models do not really specify how they do it, but rather say that or excuse me, but rather I have inferred that maybe they pick up a little bit over time, and during that overall collection period, then if they're doing very well on their affinity, perhaps that antibody they have will help them bind to the antigen more. So in addition to this, the B cell will then travel over to the T follicular helper cell. In my model, I've also kind of simplified this a little bit, so there's no traveling between an FDC and a TFH. They just all touch at once. So what I've implemented is, as soon as a B cell touches a T follicular cell, then it'll increment a clock in that cell. So the B cell only has a limited amount of time that it can touch a T cell, excuse me. And the idea behind that is, if you have a lot of B cells around a T cell, they're all trying to compete. And then the T cell is only going to reward the one that has the highest affinity. So it will choose the ones with the highest affinity to give a certain survival signal. And once the time is up, then the cell will check, do I have enough of that survival signal? If yes, then I can return to the dark zone for recycling. If not, then I have to undergo apoptosis. So very soon I want to go into a demo here, but something I also considered with my model is, by having these diffusion chemical gradients, it does impact for performance a lot. And considering this is a 2-day project, I ended up simplifying this a lot. So I started off with these stromal cells that secrete a CXL12 gradient, which Derek talked about a few days ago. It will bring the B cells back over to the dark zone when they need to be recycled. And similarly, the dendritic cells should secrete a 13 molecule. But in reality, I just have these linear gradients. So it's constant, it's always there. And what happens is, every time a B cell returns to the dark zone, then it will just stop in this area. It's just a small detail. So very quickly, I want to show a sort of demo. So I have this screen recording here. So my pale yellow cells are going to be my central blast. And those cells are going to be the B cells that always stay in the dark area. Now, after they proliferate enough times, then they'll become central sites, which are going to be in bright red. Their goal is to move over to the white zone on the right, where they'll touch dendritic cells and T follicular helper cells, which are the bright red cells there. So this brings me to another simplification I've made to my model, which is, other models may consider that you might only have like a limited number of cells that are seeded, or they have the antigen with them. In fact, I've just given all of them the identical, these are identical at the very beginning of the simulation. So they want to proliferate for a little while, and then they'll migrate over to the right. And what's happening here is you have these, all these B cells come into a clump around my dendritic and follicular cells. The dendritic cells are going to help replenish the antigen. And if several B cells are touching a T cell, some of them might actually start to die off because they have poor affinity. Whereas the ones with good affinity will start to return to the dark zone. So we just go a little further. So here's some that are returning now. So you also see that there's a significant reduction in the overall population. It was very dense. Now you've killed off a lot of the low affinity ones. As for my overall result of the model, though, I would say I may not have achieved my hypothesis. What I want to see is a steady growth of my overall affinity. So this is the mean affinity across all the B cells. It should start to grow towards 100%. Instead, it bounces around a lot. So what this shows is my hypothesis may be incorrect, and it may be that either I have to fine-tune the parameters more, or that it's the case that choosing the amount of antigen and having it affect both the B cell's performance over here, as well as the amount it can proliferate on the dark zone, may not be the right approach. So I've also created some other plots that affect, or excuse me, that show the number of B cells and how many exist with each amount of affinity. So what you want to see towards the end of the model should be, most of them are towards the 100% mark. Similarly, I've also considered several ways to record the a separate hypothesis, which is how does the apoptosis rate affect the success rate of the B cells? So instead, a different plot I have considered here is at what point does a plasma cell occur? So what is its affinity, or what is its generation upon exit time? So what I'm trying to say here is, in all of my B cells, I record a generation number. So every time there's a division, it'll increment its by 1. And what I'm trying to show is that there's a chance for B cells to become plasma cells and be able to exit the model very soon. Just because as soon as they appear, they have a certain chance, just by probability, they can become perfect right away. And I think it's better to show that over here. So if I have my affinity field will start in blue, which is poor affinity, and migrate up to bright red. So that's the best one. It turns out that you can start off with some bright red best affinity cells just by chance. And what this plot shows is that there's a pretty good chance that all of the affinity improvement I'm seeing in my model is just due to chance. So I don't want to take too much longer with this presentation, but I do have quite a bit more to go. So, oops. So the affinity algorithm I've considered is the same one shown by Dirk a few days ago. So I won't go into too much detail there. It's just checking, is this an exact match with the antigen? I also showed that there's an affinity field which measures the level of antigen in my cells. This is something I did very early on, so it worked out pretty well there. I also showed the affinity histogram. Another thing I want to show is, even after I wait a very long time with my model, then I may not necessarily see a whole lot of improvement with my overall affinity. So again, what I want is this plot to be reversed by the time the simulation ends. You start off with a lot of poor affinity cells, and then you get up to a lot of good affinity cells. So sometimes you do see just general improvement by chance as well. Here I'm showing the mean affinity across all the B cells again, but this time you can see a little bit of improvement by the end. Sometimes it just happens that way, sometimes it doesn't. And there again, you can see a bit of randomization. So to go into more detail about my parameters, I was lucky in that many of them are available online. Some of them I could take exactly as is, such as the maximum amount of time a cell spends interacting with a T cell versus the amount of time it spends, or it needs to, in order to get that sufficient survival signal. Other parameters I scaled down a little bit, and some of them were not so easy to measure, at least during the 2 days I had. There are also some randomization values that were out there, which affected the proliferation in the dark zone. So one idea here is, in that dark zone where all the B cells proliferate, they have a chance to lose some of the antigen during that process. There's an 88% chance that they will. So, or an 88% chance that it'll not be split evenly, excuse me. And sorry, I'm still misspeaking. A 72% chance that there's asymmetric division, and when there is asymmetric division, the maximum amount of antigen it can retain is 88%. Okay. At the same time, I've also adjusted some other parameters where it's a little bit difficult to express how I adjusted them, just because I ended up using a completely different algorithm than what was out there already. So that's about it. And there's some future work to do, but I think I'm happy with the results so far.
I think it'd be really interesting to compare that to the model that was shown by Derek and see how one could harmonize some of those questions. And I think it's a lot of progress, really impressive.
Thank you. Yes, I was not able to get a hold of them in time, so I ended up building kind of a caricature or toy model. But I explored different questions than they did, so there's still a bit of worth to this model.
Oh, even if you did exactly what they did, it's always worth building it a different way, because you learn something. The choices they made might not be the choices you make. And so I think it's the fact that they did some of the same things doesn't mean that it's not worth doing by any means.
Yeah, and the way you are developing your simulation, I would encourage you to do it your own way, because I think, first of all, you learn more. Second, you could come up with new ideas to solve problems others solved the other way, and yours could just be better.
Yeah, in a way, it's almost like a kind of replication to have two people working on the same biological problem, and approaching it different ways. Or I guess reproduction would be the word there, not replication. But yeah, reproduction of models is actually something that we didn't really talk much about the past 2 weeks, but there's definitely a discussion surrounding reproduction lately that we could get more into in later discussions. But it's certainly worth the time, even if somebody's already done it, to do it again. And like Pete said, he's even answering slightly different questions than the other group was. So
fortunately, Kring was not able to be here, but she was here yesterday and she helped with the basic part of the model, or like setting up the model pretty much. So what she and I were interested in looking at was how ECM-Cell interactions affected cell migration. And so between ECMs and cells cells can interact with ECM by making adhesions, and that allows them to move or migrate. What we were both looking at was collective cell migration, but I think currently it's really more it's more single cell, I would say. But you know, in terms of how cells can interact with the ECM, there's also how the properties of the ECM can affect how cells can interact with it. So like the density, then there's pore size, and then there's even bundling of fibers within the ECM. And also, you know, the cells are able to remodel the ECM fibers either through alignment or maybe by depositing more fibers, or even degrading fibers, I think through like LMPs, I think they're called. And something that or at least my lab looked at, was like cell invasion distance or cell invasion speed. I didn't really get to implement this part, but like that's something that I could do later on. So for the first part of the model, we were looking at how we could implement sort of a cell ECM adhesion-based migration. So our model consisted of 3 types of cells: epithelial cells, mesenchymal cells, and ECM cells. It was really, I know here I show, it was a 2.5D model, in that the ECM cells were a layer of ECM cells at the bottom, and then the epithelial and mesenchymal cells are at the second layer. And then the way that the epithelial and mesenchymal cells were able to interact with the ECM cells were through adhesion, through the adhesions flex plugin. And so we had set it up so that the epithelial cells were able to interact with each other through the CC's molecules. The mesenchymal cells were able to interact with the ECM cells. And so the mesenchymal cells had the CM molecules, while the ECM cells had the MM molecules, and then the epi cells had the CC molecules. And actually, I think we also ended up having the the mesenchymal cells having the CC molecules in order to facilitate further migration. And then one of the parameters that we were looking at, or that was like sort of vary, was the threshold for the epithelial to mesenchymal change. So based on this field here, which was remain constant really throughout the whole simulation, based on this, that would determine how much or whether the cells would change from epithelial to mesenchymal to be able to migrate. And then it would also determine how or what the molecular density for the CM would be, so that it would make it so that it would interact, there would be high interaction between the mesenchymal cells and the ECM cells. So so this is one of the threshold. So this was the threshold that I used. From this field, it's really from a 0 value to a 0 to 1 value. Don't think it plotted the color bar there, but so you can see here for a threshold of 0.35, the cells become mesenchymal. So, and that's based not only on its interaction with that field, but also with the medium. So right now here, you saw from the very first image that only the cells on the outer, on the periphery of the cluster, that those are the first ones that are mesenchymal, and that's because they're not only is the threshold value low, but it also is in contact with the medium cells, or the medium that's in the second layer. And then I also plotted the molecular density that allows for the mesenchymal cells to interact with the ECM cells. And then here you can see that at first, because it's from 0 to 1, that only half of the cells on the periphery are at first mesenchymal, and then that increases as the cells kind of dissociate from the cluster and allow for the cells towards the middle to become more to become in contact with the medium. And then this is how the molecular density increases for those cells. And then here the threshold is too high, and so it doesn't really allow any of the cells to become mesenchymal, and so that's why you don't see any change of the cells from blue to green. So this was using the adhesion flex plugin to be able to see how cells can interact with the ECM. But the next step, we were looking at how maybe implementing how ECM alignment could affect the cell migration. Let me see if I can go. Oh yeah. So the next step was looking to see if maybe at first we were thinking of using a scalar field to where we randomly assign a value to each cell, ECM cell. And so based on that, based on the contact of the mesenchymal cells and the ECM cells, they would be able to migrate in the direction of the ECM cell that has a higher value, so higher alignment that would be. But then we ended up changing it to a vector field, and I didn't really get to finish this part. I was able to get to this field, vector field. So you can see here, I was able to get like the random random directions for all the vectors. Well, another word, but and then I still had the epithelial cells, mesenchymal cells, and the ECM cells. And then I think for this part of the model, because it's more alignment, it's based on alignment, I think I would have to remove the adhesion flex plugin, and then maybe just base the kind of like the migration on the magnitude of the vectors associated with each of the ECM cells. And so as the cells are, because the ECM cells are smaller than the epithelial and mesenchymal cells, as the mesenchymal cells are on top, they're able to make contact with multiple ECM cells. And then I'd be able to take sort of an average of the vectors or alignment vectors from each ECM cell, and then determine the actual vector for the cell migration. And so in that way we could allow for cell migration. But yeah, that's really all I got to do. But and then, you know, thank you to all the instructors for setting up the workshop and for all the help throughout the hackathon.
We'll have the Turing group go now, last but not least.
Hi, everybody. I guess this is our turn.
Mhm.
I would like to first thank you very much to the organization, and thank you very much for this opportunity. I also want to thank to my group friends. They are all very supportive, and we worked a lot this weekend. So I want to share my screen. I think. Can you able to see my screen now? I guess I'll continue like this. Okay. Our project is Turing type of pattern formation in reaction diffusion systems, and we specifically want to observe the pattern formation in digit limb type of model for digit patterning. And actually, in the last minute, I put the simulations from CC3D to the first slide, and we'll get there. And so it's originally Alan Turing came up with the idea that reaction diffusion systems might be responsible for the emergence of pattern formation in nature. Its range is very large, from very wide, from patterns on animals to the animal plant development. And we would like to observe the pattern formation from the lens of Turing type of pattern formation using the CompuCell 3D environment. So we also wanted to, our ultimate aim is to observe the digit patterning. Okay, actually our study, we would, if you are interested, we are inspired by this article you can see. And the here is the shape, here is the biology that's behind we would like to model. And okay, and the mathematical model, actually, this is the, sorry, I get a bit excited. This is the pattern formation we would like to make ultimately, but we started with the well-known Semiconductor model, and this is the mathematical model that we considered. We wanted to start with it because we already had some demo code on it, and we already have a lot of information about Semiconductor model. So there are some limitations on the model that we considered while we are constructing our model into CompuCell 3D, that the diffusion, there is a mechanism between the inhibitor and activator. So there are two species here. U is the activator and V is the inhibitor, that we want for patterns to emerge. It's a well-known concept that long-range inhibition and short-range activation is the necessary requirement so that we can have patterns. And this is the model that for the digit development. And if you are interested, again, we are inspired by this article, and this is the scheme that we would like to consider. I want to go back to the plan for 2 days, and we couldn't able to finish all of them. We are planning to continue to get to our ultimate goal. So we wanted to see Schnackenberg kinetics on a cellular level, and our 2 important steps we have, the secretion and uptake. And you want reactions happens only inside the cell, and diffusion is happening everywhere. And then we want to, once we could able to construct our reaction diffusion model in CompuCell, we would like to adopt other kinetics. That's what we learned, and we would like to adapt to other kinetics. And then we also, it is necessary requirement for the digit development that pattern formation and cell migration, these are the important concept of cellular mechanisms that we would like to include them. So eventually we could able to get the digit patterning. So far, I will actually need to thank our group friends for the amazing effort that they put, and I might need help at this stage. So this is the XML file that you can see, and we have the cell types and the initialization concepts that you can see. And the main things are done under Python steppables. And Ross, would you like to take it over?
Yeah, sure.
Yeah, thank you.
So quickly, I too would like to thank both my team and the hosts for this support. It has been amazing, really. I will get on to it. So here you can see our equations. So you can see that there is a term in the second equation that looks a little funny because we are subtracting I and then again adding I. We can take you to it through the code. There is a reason for that. It's a trick to have an uptake term. So the minus A and the minus I were interpreted as an uptake term from the cells. And the minus I wasn't there. So we had some trouble uptaking the chemical inside the cell, having a part of the equation to correspond to that. And we did this little trick so that we wouldn't change the equations. We would still follow as close as we could the Schnackenberg equations, but we would create that term where we needed it, without measuring the fields before uptaking the chemicals. So you can see the parameters we focused on. These are known to give rise to patterns in the raw Schnackenberg equations. So you can also see the trackers we were loading to check the uptake, as you see in A in and I in for the 2 fields, and the secretion in the out terms. So let's move to the next part. Yes, so here is our stepable. Almost everything happens here. We have the diffusion solver to do the diffusion outside the cells. We don't want to mess with that inside the cells. So we have the rest of the equation without the diffusion as a reaction of the cell to its environment. So here we can see that, can you go to the next one? I think the next one has the comments as well. Yeah, this. Okay, so what we are doing here is that in the first Monte Carlo step, we are doing a secretion from the cells inside the medium, and you can see that we are dividing by cell volume. So this was something that it took us some time to realize, that the way that the secretor works when it secretes inside the cell gives this term that we put inside the parentheses in each voxel of the cell. So when we wanted to output an amount of, let's say, 0.5, and we put it in the parentheses, we wanted to output it for the whole cell, but it did so for each voxel. So it gave us a huge amount because we had about 100 voxels per cell. So we corrected that by dividing by cell volume. And you will see other terms that also have this division to correct that. So after the first step, of course, we have the uptick. You can see a randomizer at first with the if that was put there in case we want to have just some of the cells do the process in some steps. So we give it some stochasticity. Now it's been put to 0, so we have every cell reacting the way we have coded it. So here we have the uptake for each cell. I won't go through the specifics unless there are questions on the amounts we are uptaking. And then we are calculating the output, what we want our cell to secrete and then give out to the environment. You can see that we are putting the values in the dictionary to have this the field tracker display them. So something interesting was that we noticed after getting what we want from the simulation that if we show the field tracker for the output of the cells in both fields, we kept seeing all the cells secreting the same amount, which couldn't be true, and we were getting the patterns we wanted. So we realized with help from Gilberto, thank you for that, we realized that there was something with the tracker, which when the difference between the secretion of the cells between the values was in, I think, the 8th or 9th digit of the decimal, the tracker couldn't display it properly, so it showed the same color for all cells. But to figure that out, because we were worried that there was something wrong with our code, we did a log file and we printed everything there, and we saw that the values were different. And the difference might be very low, but we checked that it was what we expected from our equations and parameters. So this is probably fine. So after that, you can see that we secrete inside the cell.
Sorry.
Yeah, that's all really. And the code now seems very, I don't know if we can say simple. It's much fewer lines than we started with. A few hours ago, it was really a mess. But I think we learned a lot, and we can't put that really into a quick presentation. We went through the system again and again to be sure that we understand the biological process, that we understand the math, and that we are translating it right computation. And we saw some very interesting things on what you can and can't do in a computation. Let's see the video.
Video. This is the initial.
Yes, this is the initial before the first secretion.
I can't
Here
play
is the video,
video.
and we can show the pictures later.
Can you see it's playing now, or it's playing? Okay.
So the video was recorded not with the uniform initializer we put for random initial conditions. We actually made the cell secrete 0.5. Every cell secretes 0.5 of the activator chemical and 0.5 of the inhibitor chemical. So this simulation was run with exactly the same thing for every cell. A uniform secretion of exactly the same amount of chemical. And despite having such uniform initial conditions, we can see that the equations, as close as we can get to them with agent-based modeling, give rise to patterns. So one interesting thing is that we know that the Snykenberg equations give rise to patterns, but if we see it mathematically, this happens in the equations in every point in space. So what is interesting that despite changing that and putting the process inside cells and not, for example, in the medium between them, we can see a rise of patterns even using the same parameters, despite changing the system. So I think that is really interesting. So in the future, on this model, we would like to check some different parameters. We are still checking that the system does exactly what we want it to do, and we don't have any errors, either logical or what we have in the output. And our goal, and we have started some work on it, is to move to digit pattern. And this is where the fun begins. It's a much more complex model, but we think we can adapt what we have learned to trying that out. So I will leave you with Melanie to tell some final things on what we want to do with digit pattern. Thank you.
Thank you so much, Lampros. And this is also a final state of our simulation. Yeah, actually, Melanie has things to say. After Melanie, I have also some little concerns that I want to share with James. Melanie, would you like to share your screen, or do you want me to talk on these shapes, or I can skip?
I mean, it's just that, yeah, so I'm working in parallel with Flores and Gulsamai and the rest of the team on the model that we want to simulate digit patterning. So it's, yeah, actually I can share my screen maybe.
Yeah.
But like the screen that you've just seen is a little bit of the output. It's not the right one. Like there's a lot of problems with it, but it's a starting point. So this is, oh sorry, this is the model that we have. So these are just declaring like a bunch of parameters, but then it mainly involves 4 variables, B for BMP, R for its receptor, the formation of their complex, and a regulatory, like a protein that acts as a positive feedback regulator for B. And these are the equations for it. And yeah, we're basically using Lampros's code as the skeletal structure of it. And here I'm just starting off by declaring like the fields and stuff. And I'm just gonna very quickly breeze through it, but we will be using, like putting in, like detecting how much field is around each cell, and using equations to determine how much of those variables will then be produced by the cells, and it will affect the field, and we will just loop that. And the pattern that we've just shown you looks something like this. We are still encountering a little bit of a problem where it's a quick fix. It's just that I forgot how we can put the chemicals out of the cell. But like if we look inside of the cell and not consider the outside interaction of those chemicals yet, this is kind of like the pattern that we are seeing inside of the cell. And ultimately, this window here with interaction of like chemicals outside of the cell, we want to see a pattern that resembles digit patterning, where there will be like spots of spots formed, which would be reflected by the complexes formed among the cells. And ideally, like what we think we can do is that we can put in some sort of like maybe cell type switching so that the digits can extend into fingers or like digits.
As James suggested, we would like to work on some other reaction diffusions, and there are already really nice demo codes provided for us in the workshop, including other reaction diffusion models like chiral Maynard, for example, is one of the example. And I just run the existing code and to see what's happening around the educational demo code. And what I observed it was really, as we can expect it with Schneckenberg model, we already worked on it with James as well. And what I was just wondering, maybe this is not a big deal, but I was wondering if this is something that we can think about it, because this is another reaction diffusion model. Let me show you. This is Fyffe-Nagumo model. And yeah, this is after some time, the simulation for the inhibitor, actually, we can see some negative values. It's like keep changing between the negative values and positive values. And I was wondering if there is any issue about it. I was just didn't feel very comfortable, so I just wanna discuss it with James. And could it be something important? I don't know. And it looks like there is a very little change in it, and maybe after 6 digits or something.
I'm sorry, what? Well, so it's possible some of these models don't work. They're quite old. They may not work. So it's certainly possible that that they're not implemented correctly.
Do you think it would be all right if we play around the parameters? Because for Schneckenberg model, it was really beautiful and it was what we expected.
I'd be delighted if you found what was wrong with them and fixed it.
Yeah, I would love to play with the code. I would love, because I was trying to do it, and because we wanted to put put the equations and put the models into our new kind of coding, and it's also in our agenda.
Absolutely. I mean, all of this material is open source, so you don't have to ask permission. You could just use it.
Thanks.
Yes, and it would be great to
talk to you, and if you fix something that's mistaken or make it better, that's great. So we're delighted that you're interested in trying to understand if there's something not working with that code.
Oh, I even didn't consider if it is not working, because we have the simulations very nice as expected. So, but the negative things that just put some question marks into my head. So I want to just share it with you. And thank you very much. I love this workshop, and I hope that I really feel like it's not enough for me for just 2 weeks, and hopefully with the group we would like to continue to finish our modeling. And it's been an amazing group, and it's also I'm very happy that I joined this workshop.
Well, thank you. That's a wonderful feedback. We appreciate it. We know we could always do better, and so we appreciate compliments, and we also take criticisms and suggestions very seriously. So we do appreciate everybody's hard work. This has been a long 2 weeks, and we thank all of the volunteers who helped teach, and we thank all the volunteers who came and worked such a long time with us and helped make this workshop a success. And I guess I should close out by saying we thank our funding agencies for providing the resources to do this.